So this is the last part of Chapter 8. It is Section 8C. Now, this last part is going to be discussing viruses. And you remember that early on when we were discussing characteristics of life, we looked at, would a virus be considered alive? And, when you think about how many characteristics does a virus have is regarding reproduction, growth and development, energy use, order, and cells. Well, it turns out that we may have mentioned this before, that a virus is not actually a cell. And that's probably one of the major reasons why most biologists do not consider viruses living, because they are not made up of cells. What a virus does, it has no cells of its own, and it can't reproduce on its own. And remember, these are two defining characteristics of life. For a virus to reproduce, it must infect a living cell and then direct the cell's internal machinery to make more viruses. Now, viruses have fairly simple structures. Think back to the cell. Even a prokaryotic cell, right, has a plasma membrane, has DNA, has, lots of different structures. Viruses basically have a nucleic acid. It can be either DNA or RNA. It has a capsid, which is sort of the outer coat of the virus, and it's made up of one or a few different protein molecules that are repeated in kind of a regular pattern. Then there's typically some type of recognition spike. Each virus has one or more proteins that protrude into the environment and is capable of binding to proteins on the outside of its host cell. So we're going to be discussing a few different types of viruses. And the first one we're going to discuss is called a bacteriophage. Sometimes it's just shortened as called a phage. And that is a virus that actually infects bacteria. And once a bacteria is infected, the virus can enter one of two cycles. It can enter a lytic cycle, where the viral DNA replicates using the cell's machinery, and that results in the death of the host cell, the bacterium. Or it could enter a lysogenic cycle, and that's when the viral DNA inserts into the bacterial chromosome, and the virus can remain dormant for long periods of time and then come out later. And here you can see a comparison between the lytic and lysogenic cycles of bacteriophage. Now, viruses are capable of infecting a broad variety of animals and plants, and viruses typically are specific for a particular host, although they can move across species at times as well. So, for example, the influenza virus that we know causes flu, that affects human cells. Tobacco mosaic virus, that is a virus that infects plants. And you probably know that HIV is a virus that basically destroys the human immune system. And with that results in the disease AIDS, which is acquired immunodeficiency syndrome. HIV is what's known as a retrovirus. It has an RNA genome, so the nucleic acid it has is RNA, not DNA. It targets cells of the immune system, and it spreads by sexual contact or by sharing blood products. And here down at the bottom is a sort of a representation of the HIV virus. And the red you can see is the RNA part. So HIV relies on the action of a very specific enzyme called reverse transcriptase to reproduce. So to reproduce, HIV uses this enzyme to convert RNA into DNA. Remember, we normally talk about in the genetics unit going from DNA to RNA to protein, but retroviruses with reverse transcriptase can go from RNA to DNA. So during normal DNA expression, the host cell will then make new viral RNA. And the HIV virus essentially hijacks the cell's own DNA expression system. So here's the life cycle of HIV. So you have infection, that's here. And then you have, here's the viral RNA. From the viral RNA, with the help of reverse transcriptase, it can make DNA. So now you have double-stranded DNA that originates from this viral RNA. And then what happens is this newly created viral DNA can actually enter the nucleus. That's what's happening here. And it can actually incorporate into the human chromosome. So literally within our chromosome, when you have an HIV infection, you have viral DNA that has inserted into our chromosomes. And that's one of the reasons why like with an HIV infection, people could be infected and not be sick for, a long period of time, and then all of a sudden, we'll get very, very sick. And what happens then is this DNA, viral DNA, you could make viral RNA from that, and that's using the cell's machinery, our normal machinery from our cells to make viral RNA, and then basically from there you produce what's needed to build new virus particles, and they are made and they assemble, and then they leave the cell, and then they go and infect other cells. So that's basically HIV. I just want to mention just a few things about SARS-CoV-2. So SARS-CoV-2 stands for Severe Acute Respiratory Syndrome, coronavirus 2. Okay, and it's named coronavirus because of these spikes here that kind of look like a crown. So the SARS-CoV-2 is part of a whole family of coronaviruses. There are other coronaviruses that cause these respiratory infections. And then COVID-19 is actually the name of the disease that is caused by SARS-CoV-2. So SARS-CoV-2 is an RNA virus, so the viral genome is made up of RNA, and then of course it has the protein coat, and then it has these spikes. And these spikes are used to bind to proteins on human cells that then allow the virus to enter.